So let me show you an example. We are studying in one project, we're studying what are the cell dynamics or during tumor growth. And the previous information that we have, it was taken from fruit flies in the larval stage. Why? Because doing imaging tumors is not very easy. So one of the best ways to do it is using fruit flies in the larval stage in something called the cephalic complex. And however, so what we were doing is they genetically induce tumors. So they label them with GFP. So you can see the tumors grow and you can start analyzing the cell dynamics. Yeah, so this is, for example, a Nature paper that they were trying to do that, and you can see when in a normal tissue, nothing happened. However, when you express these two proteins, RAS-B12 and Scribble, in the mutant form, you can you get these huge tumors. The problem is that this technique is not very efficient because the larvae, when you're trying to image it, is moving. So you have to make very short movies. However, in the pupal stage, it's much easier because the pupae is not moving. So what we are trying to do is trying to image the early stages of the pupil development, and what we found is that the tumors start detaching in small pieces. So let me show you very quickly. So look, what they have. This is the normal pupae, and this is a movie that show the development of the leg and the wings. So let me show you what you're seeing underneath the cephalic complex. You see here the legs in blue. You see the wings in red, and why you don't see the other wing? Because it's in a position that it will be located here. Maybe you cannot see it. Here you find this is a boundary that we have underneath the legs. And what they are doing is inducing these tumors. And when we induce these tumors, you find that pieces of tumor start circulating. Yeah, you find green spots everywhere, and they start moving following different trajectories. So the question is, do these tumors can go from one side to another side? And this could be an example of metastasis or not. So using this project, we can ask different questions. Yeah. So what I want that you do, these kind of people can, or what I want that you do, if you're in this project, for example, you can ask, how similar are those moving tumor spots? Let's call it like that. And you can ask the size, the velocity, the trajectory. But why you ask this question? Because maybe the characterization of the cell dynamics can allow us to infer if these moving tumor spots are alive or not. Or maybe they can allow us to determine if they can produce metastasis or not. You can ask another question like, what is the average size of the tumor size spot during development? And why? Because maybe if they start growing and growing, or you can see them divide, you can say that you can think that they are alive, or maybe if you see them that they start breaking into small pieces, you can look into literature and say, oh, maybe this is a characteristic of apoptosis. Yeah, so trying to answer what I want is that you ask questions and explain why you think that those questions are important. I don't want that you don't have to write 20 pages, just something, an outline, so we can discuss. Another example of a question is, do you see those spots displaying cilia or flagella or protrusions? A protrusion is when the cell have an actin elongation that allow them to move. Yeah, so cilia, flagella, or protrusions. These protrusions are like fingers that allow cells to move. Can help us to understand if the mechanism, if they are moving, how they are moving. Maybe they can move because they have protrusion or because they have flagella or they have or actin structure. And of course, and a question that you have to ask is what other people have found in this system. For example, we know that people using tissue culture, when they imaged legs, they found dots, and we can try to see if these dots are similar to those one or not. And what I want is that, for example, this question is amazing, to ask why those questions are important, and then try to see if those questions are testable. For example, the first question measures size, velocity, and trajectory. You can do it manually with ImageJ, or we can install a plugin to measure the average size during development. Yeah, you can take the during the movies first image, second image, third image, and see if they start maybe growing. To see the cilia or the flagella, you can start zooming in. Yeah, this will be very easy to do. And maybe to verify that, you can write that for a future research. So yeah, you can say to verify, you you look for, you can suggest a marker for this structure, like an staining, a way to stain those things to see if what you see is actually true or not. And there will be questions that are amazing, but they are very difficult to test, and we want to that you identify them right away in the first week, rather than try spend the entire semester and realizing that maybe there is not an easy way to do it. So remember, make a list of 9 questions, and this can help you to know how to phrase the questions in a better way that I did, in a more scientific way. However, for this exercise, the outline is very casual. I just want that we have a discussion about what kind of things you want to do. If you have any questions, just let me know, and have a good day. Bye.